and make sure that the soil is nice and moist. If you're germinating seeds, those seeds have to stay evenly moist, otherwise they'll abort their germination and die, and then you got to start all over. So the greenhouse, if you're in there, make sure to be aware that the sun beating down into the greenhouse is definitely going to warm it up, even though it's kind of cool outside today. So keep an eye on that. Did Duncan talk to you about the idea that there are monitors here on the weekend? If you have to get in to water your plants on the weekend, you can do that. They have the monitor system. Did he talk to you at all about that? So, okay. So if you water, like Thursday during your lab session, you take care of your plants, but if the weekend is full on sun, even if it's cool outside, it's going to get hot in there. Even with our swamp cooler going, it can get into the 80s and 90s, especially if the outdoor temp is mild. And then we have monitors that are here on Saturday and Sunday for a select period of time. I'll send an announcement of what their hours are. I think it's something like Saturday 11 to 2 and Sunday 1 to 3, something like that. And then they can let you into the greenhouse and water your plants. So they are located, usually they have a whiteboard that they'll put out right outside the zoology museum, right as you walk in the door closest to the library. If you go into Weston there, you see that wolf in the glass cabinet. That's right where they put a whiteboard, and they'll say who's on duty, phone number, and you call them if you need to get into the greenhouse. All right. So I'll put that in an announcement so you have that information. Okay. And we are looking at some strategies that different plants have depending on where they're native to for improving on the C3 cycle, mainly improving on Rubisco. Rubisco is kind of the weak link there in the sense that if it sees not enough carbon dioxide, but there's enough oxygen around, it'll just go ahead and take oxygen into its active site. And so in areas that are consistently warm every day, pretty much, and periods of drought, you could see where the plant might choose to close down its stomata under those conditions because it's getting dehydrated. It's water stressed. And so that's one of the first things it'll do is close down its stomata. Well, then you have no carbon dioxide coming in. And then you have a situation where Rubisco doesn't see enough carbon dioxide. And then oxygen will win and come into the active site, and you'll end up with the PCO cycle kicking in, which is complicated and expends energy and actually gives off carbon dioxide instead of fixing it into sugars. So not a good thing. So some of these plants that have adapted to these consistently warm conditions around the equator, typically they can do the C4. So we looked at that and we went through that in pretty good detail, looking at the spatial difference of where things are happening, and that the bundle sheath cells, when you look under the microscope, they're nice and plump with these plants. They have lots of chloroplasts. They're very active. And that's where the bulk of the C3 cycle is happening. And for sure, that's where Rubisco is located. So they've tucked Rubisco into the bundle sheath cells. And the idea here is that in the bundle sheath cell, they've also separated out spatially, like the light reactions really aren't happening in the bundle sheath. They let the other cells do that in the leaf. So that way the bundle sheath isn't cranking out a bunch of oxygen, because when you run the light reactions, you can't help but get oxygen as one of the products. And so the bundle sheath really doesn't get a lot of oxygen. But even so, if you start closing down your stomata, you can run into an issue. But with these plants, because they have this C4 idea tacked on, kind of woven into the start of this process, they'll take CO2 in, and instead of interacting it with Rubisco right away, they do the peptase enzyme instead, which is super picky. It only likes carbon dioxide, and it only will take that and the phosphoenolpyruvate into its active site. which is great because then you'll get OAA, which gets converted to malate. And so you can think of malate as the vector. It has CO2 as part of its structure. It's going to move that CO2 into the bundle sheath, cleave it off, and feed it to Rubisco. And then the other product is pyruvate. Pyruvate shunts back into the mesophyll and gets converted back to the starting PEP. But there's energy involved in that step. And so they have to expend some extra ATP, and NADPH as well is involved in this cycle. So there are costs, but the idea that the benefits outweigh the costs. So if you're a plant native to this warmer habitat with periods of drought, it's a win. It's a good thing to do. And then we also looked at, okay, let's push it even further, where you have the desert environment. So now we're talking about the CAM pathway, crassulation acid metabolism, named after the family Crassulaceae, which are succulent plants. But now we know other plants can do this. But basically, we're looking at a very unique habitat where you have blazing hot days consistently and long periods of drought. You have the monsoon season, but it's long stretches of time where you have drought. And so now if you open your stomata in the day under those conditions, you'd shrivel into a crisp and die pretty quickly. So they don't choose to do that. They have evolved to go ahead and wait until nighttime. That's when they're going to go ahead and open their stomata and take up CO2. But they can't run photosynthesis at night, certainly not the light reactions, and certainly not, they can't do the Kelvin cycle because those enzymes are light activated, many of them. So that ain't going to work. So then they go ahead and just store it. They'll take that CO2, stuff it into their vacuole, keep doing that all night long, get a nice buildup of malate as their storage form of CO2, get that into the vacuole. And then during the day, close the stomata down and go ahead and start running the idea of getting the CO2 to the Kelvin cycle and allow that to happen and cook along, which is great because then they're going to get their sugars. So that's where we left off. We're looking at on the left where you have the dark and the CO2 coming in. What's nice is it's a lot of the same players as the C4 cycle. So you don't have to memorize completely new players. You have that same peptidase enzyme reacting with CO2 and the phosphoenolpyruvate. And then it gets converted to OAA like we saw already with C4, and then to malate. And then malate is stuffed into the vacuole. So a couple of things during the night here. You see in the chloroplast, you see starch. Okay, so when a plant has enough sugar and wants to save up for a rainy day, it typically will store sugars in the form of starch, which gets stuffed into the chloroplast. So that's actually the source of phosphoenolpyruvate. Okay, so PEP is actually coming from the breakdown of starch. So I would like you to key in on that. Okay, and then during the day, you can see malate coming out, and they're showing how CO2 eventually gets to the vacuole, to the chloroplast, sorry. And the Rubisco is sitting in that chloroplast, gets bathed by all that CO2 getting cleaved off of malate all day long. And so this is the same cell, even though they're showing, two cells here, but really it's just day versus night. So everything's happening in the same cell. You don't have that spatial segregation with a bundle sheath and a mesophyll cell idea. You don't have that. It's all happening in the same cell. But it's a lot of the same players, a lot of the same enzymes that are running this pathway as well. Okay. Questions on the CAM or the C4 pathways? Okay. So CAM plants, there's a handful. They're still looking for more, but so far there's only literally a handful of CAM plants that they know are what we call facultative. And this kind of implies it's a take it or leave it idea. So what happens is if a CAM plant is facultative, and if it's getting, let's say it's the monsoon season, and so it gets just all this great amount of rain, and it's not under a drought currently, it could decide to turn off the CAM pathway and just run the C3. So if you have maybe a house plant that's a facultative CAM plant, if you are generous with your watering and always are making sure that it gets enough water, then it could go ahead and shut that down and just run the C3 cycle. But then if the monsoon season leaves and you get a long period of drought, or you go on vacation and forget to water your plants or don't water your plants, and they start to get water stressed, then they can turn back on the CAM pathway and get that up and running. So it's a very nice idea because it's, if you don't have to run the CAM pathway, it's nice to be able to turn that off because of the extra cost to it. And then just go ahead and run the C3 cycle. So it's a very nice strategy. But like I said, only a handful of plants that they know of so far are capable of doing that kind of option. So they've looked and they definitely see many, many plants in the family Crassulaceae. I just listed out a couple of genera there that are capable of doing the CAM pathway, but then they started looking at other angiosperm families outside of Crassulaceae, and they found, oh yeah, there's a bunch of others that can do this too. And it's not like everyone does it that belongs to that family. And so it's kind of all over the map in terms of who has the ability to do this. And then you can look at certain families of plants, the angiosperms, certain families, and you see a whole mixture. Some of the species that belong to that family only do C3, some do CAM, some do C4. So it's kind of all over the map. So some CAM plants that are maybe something you're more familiar with, a variety of cactuses, cacti, euphorbias, pineapple, which is a big agricultural crop, vanilla as well. Vanilla is actually taken from an orchid that does CAM. Agave, what do we get from agave? Yeah, $1 billion crop right there, tequila. And so they do CAM, and a variety of orchids do that as well. So orchids, we do have orchids this far north. They don't do CAM. We're talking about orchids that are more tropical and in consistently hot environments. Most orchids belong on more on the equatorial line and south of the equator. CAM plants can also be found in what we say are more primitive. So what we mean is going out of the angiosperm group, out of the flowering plants, and going into the gymnosperms. So there are a couple of gymnosperms that can do the CAM pathway. So the CAM pathway, because they can close their stomata all day long, they win in terms of comparisons, in terms of water loss. They can conserve a lot of water. And so 50 to 100 grams is lost per gram of CO2 fixed. Compare that to 250 to 300 for C4 and 400 to 500 grams of water lost per CO2 fixed if you're just a straight ahead C3 plant. So yes, CAM plants win for sure. And when you look at the evolutionary process of these different adaptations, like I said, all plants do C3. So that evolved first. So actually the C3 cycle evolved in bacteria, cyanobacteria. And so they did it first, but then plants that started to colonize land, algae, of course, do the C3, and then land plants. So C3 is first. And then because they know that CAM plants can show up in the gymnosperms evolved before the angiosperms. So the CAM plant mechanism came next in the evolutionary timeline. And then the last specialization, most recent one, is the C4. So they're very interested in the C4 idea. There are groups of scientists that are trying to engineer plants that can't do C4 to start doing it. They've not had success with that yet, but that is something that they're looking at. They're also looking at the CAM pathway more intensely with climate change issues looming, and certain areas are getting hotter, and crop production it's becoming dicier for certain crops to deal with that. So they're wondering if they can learn from the CAM pathway and those plants and see if they can come up with some strategies to help. So they're still looking at these cycles to try to deal with, especially focusing in on climate change issues and crop production. So all of these, the C3, the CAM and C4, I mean, basically all these plants are going to run the C3. And the goal of the C3 is twofold. It's to get sugars available, but also to regenerate the RUBP to keep the cycle going. Okay, so the C3 cycle has to keep zinging around all day long, but part of that is to get your sugars. So when we look at the C3 cycle, what we're really zooming into now when we think about sugar production is once we get that pool of G3P building up through the process of the Calvin cycle, then some of that gets siphoned off to make sucrose. Or some of it will get stored up in the form of starch. So that's what I want to talk to you a little bit now. I'm not going to go into huge detail on the physiological pathway, but there is a couple of ideas to think about. So if the plant has plenty of sugars going on, like, okay, I'm really cranking out G3P, I'm making a lot. I have the ability to make a lot of sucrose. In fact, I don't really need to keep going so strong with sucrose production. I'm going to save up for a rainy day. I'm going to divert some of that for starch. Most plants do that. There are a handful of plants that don't do starch storage, but the vast majority do. So if that's the choice, they'll just keep the G3P in the chloroplast because that's where the Calvin cycle is cooking along. So they'll just keep the G3P in the chloroplast and divert it into starch production. And then they'll just keep the starch in the chloroplast. Like I said, a handful of plants actually will take the sucrose that they have extra and they'll stuff it into their vacuole, but that's a minority. The vast majority do starch. The reason I'm mentioning it is there's a couple of big crops, oats, wheat, and barley, that those are phenomenally popular crops worldwide. And so those are known to do sucrose storage instead. But okay, so let's say the plant says, nope, I need to keep making sucrose. I need to do that because I need to feed developing tubers, new leaves, maybe there's some flowers forming, got to feed all of those different parts of the plant body. So then the G3P goes out into the cytoplasm. And it's actually the cytoplasm where you're going to find sucrose getting synthesized. And then, of course, then it can go through the plant, through the phloem translocation idea. and go from source to sink through the plant body. So if the leaves, it's typically leaves that are doing the photosynthesis and making the sucrose, and then the sink tissue is getting the sucrose. So they can use it right away for metabolism. You know, sucrose is excellent for that because they can run it into glycolysis and start breaking it down and making the situation where they can get their ATP needs met. They may have to do some maintenance. They may have to repair some damage, and so they may have to build carbohydrates and help repair damage perhaps, or just part of the growing process, laying down new tissue, or either the leaves, the source tissue, or the sink tissue could store it up and convert it to starch. Because they're getting lots, they have lots of sucrose, and so they'll go ahead and store it up. So right now, with all the leaves turning, with the deciduous trees and shrubs and so forth, a lot of what's going on is the sink tissue is getting the sucrose drained out of the leaves, so to speak, and is getting stored up as starch all for, you know, all fall. That's been going on, and then it'll stay that way through winter, and then it will mobilize in the spring to give the plants a jump start in the spring for growing and becoming active. So the idea here, when a plant is deciding, so to speak, what should it do, starch or sucrose, it really is an either or situation in a single cell. So a single cell is either going to make starch or it's going to make sucrose. And there is kind of a bottleneck process here. So this little translocator, they call it, that is a transmembrane protein, transport protein. And what it's doing, it's a antiport protein. Anti meaning things are going to go in opposite directions through it at the same time. So what you see here is the players in this are phosphate, inorganic phosphate. You see that going through the translocator, going into the chloroplast. And then you see the triose phosphate coming out of the chloroplast through the same translocator. So I'm not going to go into big detail on this other than to say that's kind of the bottleneck, and it is tightly regulated in terms of, is there enough of the inorganic phosphate to come in so that a triose phosphate can come out or not? And so it makes sense from a plant's perspective that this decision to make starch or sucrose is pretty tightly regulated. And so that is one of the regulating processes. You have to have a phosphate going in order to get the G3P coming out, which is actually in the form of DHAP. But anyways, you have a one-to-one transfer, antiport process. Then this pathway that you see in the cytoplasm, the cytosol, you see the DHAP coming from. So the triose phosphate is G3P. When it gets out into the sucrose, it gets converted to DHAP, dihydroxyacetone phosphate. And then you can see there's this whole pathway out here, but then you finally get sucrose. I'm not going to go through it. Just recognize that there's a couple of enzymes in this pathway that they know are tightly controlled. Again, so there's more than one. There's certainly that translocator as a point of control, but then there's some other enzymes here that are under control. They're not really showing that in this image. Just take my word for it. And then the key thing is that you have sugar production in the cytoplasm, starch in the chloroplast, and that decision is tightly controlled. And one of them is this translocator protein, where you have to have that antiport process going on. So if there's not a lot of phosphate out here in the cytoplasm, if there's not a lot of phosphate out here, that means that you won't have a lot of phosphate going in. So you won't have a lot of triose phosphate coming out, which basically means it's telling the plant, make starch, go ahead and make starch. On the other hand, if there's a lot of phosphate out here and you have lots of phosphate going in, then you're gonna have a lot of triose phosphate coming out, and that's gonna favor sucrose. So it's kind of this either or situation in one cell. Okay? Any questions on that idea? So looking here, this triose phosphate in the, as part of the Kelvin cycle, that's your G3P. Coming out as DHAP, I just wanted to point out when we go back to this image that we've been looking at with the Kelvin cycle, the what we're talking about is right here. They're displaying it right here. That last G3P on this image converted to DHAP and export. So that means it's going out for sucrose. So this other one here, starch, you can see if the signal is, no, keep the G3P in the chloroplast, don't send it out, then another option is it's going to go ahead and make starch. Okay, now the next series of slides are for study purposes only. It's in case your brain likes to look at things this way. It has tables of different reactions that we've covered. So if this helps you, great. Otherwise, don't worry about it. All right, these tables, there's a series of tables here, and it's just if you find it helpful, go ahead. But all of this stuff we've covered in the all the other PowerPoints. So this is more just for your own study purposes. Okay, so there's several slides here. So use them if they're helpful. Otherwise, don't worry about it. Okay. So we have covered a lot in this slide set, plus that whole slide set on the light reactions. So please keep up on your studying. This is a lot. And I just want to caution you, if you wait until, our next exam is, we're in week 9 right now. The next exam is week 12, the week of November 11th. But this starting, the light reactions are going to be on it. So will the Kelvin cycle and PCO cycle and C4 cycle and CAM. That's a lot. And if you wait too long to start solidifying that, I've seen in the past students mix them all up, and it's just really sad. And it's a bummer all the way around. And so this does require repetitive studying, looking at this material, thinking about it, working with it. When I had to learn stuff like this, I wouldn't do it at the library because I looked like a weirdo, but I would act it out. I would get up and move around, and I would use things in my room to represent certain molecules or chemicals, and I would talk it out loud, walk around and act it out. And because just reading it didn't work for me. So try to find a way that works for your brain, but detail, I am going to be looking for detail. So the way we've talked about it and looked at it in class sessions, that's the level of detail I'm looking for. So if this is my plea to you to please start studying this. If you haven't already, don't wait. A little bit every day. You don't have to go hours and hours. Just, take 20 minutes to a half hour to go at it and then take a break, do something else. But if you do that consistently, it's amazing how much that can help transfer things into your long-term memory. That's where you need it on exam day. You need it in your long-term memory. Okay. All right. Is the next slide set available in EduCat? It's not there. Eek. Let me get that. And let me make sure. All right. I'm going to need to do this. Okay. All right, if you refresh, you should see a PowerPoint called Signals from Sunlight. So that's the next topic. All right. So we've just left the huge topic of photosynthesis. But we're still going to think about light from a slightly different perspective now. So when we were looking at the light reactions, we were thinking about the idea that primarily chlorophyll A and B are absorbing light energy. And when that happens, that energy gets funneled to a special chlorophyll in the reaction center. And that absorption with that special chlorophyll excites an electron. And then that electron goes through that electron transport chain process. Great. Okay. Now we're going to look at the idea that you have light energy being used more like a signal than it is kicking off an electron transport chain process. Okay. So now we start to talk about photoreceptors. And if you are a plant, certainly light is necessary for the light reactions of photosynthesis and is hugely important, but it's also hugely important in the sense that they use light in other ways to assess what's going on around them. It gives them very vital information that they need so that they can, if they have to, they can change their behavior to better adapt to what's going on around them. So that sunlight coming and striking their plant body, they have a variety of photoreceptors that will absorb light energy, but instead of like with the light reactions kicking off an electron transport chain, it's used as a signal to tell the cells and tissues, tell the plant to do something, change its behavior in some way. And so they actually, these photoreceptors will actually do something kind of simple. They just change their shape. And that shifting of their shape allows them to have that signal sent into the cells, ultimately to the DNA, and tell the DNA to alter the gene expression. So we talk about signal cascades under this condition, where the plant is gaining information from outside of itself, so sunlight and quality of sunlight, amount of sunlight, and then that signal gets sent all the way to the nucleus, to the DNA. So it's a signal cascade. And that's not unique to plants. You're doing that right now as you sit there. Your body is doing this too. And so the fancy word for the signal cascade and the response that will happen is called a photoresponse, which kind of makes sense since it's due to light. But basically, they're absorbing photons of light, but it's not kicking off an electron transport chain. Instead, it's acting as a signal. And these photoreceptors typically are protein, but then it also has a non-protein portion to it. that they call the chromophore. And that's the portion that actually is absorbing the light energy. So the protein's really not doing that. It's the chromophore attached to it that will absorb the light energy. And then the whole thing, the protein and the chromophore, they'll shift their shape. So just that change is enough to then get that signal cascading into the cell, ultimately to the DNA, and tell the DNA to shift their gene expression in some way, hopefully to better work with what's happening around the plant. Lots of active research going on with this. This is, so there are still some things that are unknown, but I'll tell you what we do know so far. Here is an interesting phenomenon that plants can exhibit. And if you take seeds and, wet them up, put them in your paper towel, wet paper towel, and put them into a cupboard, and then you forget about them. When you unwrap the paper towel, you might see something like what you see on the right. All right, you just force the plant to go through etiolation. So if you have, I've done this in intro botany class, where you put some seeds into a pot like that and stick the whole thing into a cupboard, water it nicely, but just keep it in the dark. And they will do this consistently. So what's happening is the seeds have germinated. They're not getting any signal from sunlight. In essence, what it's telling them is, I am getting totally shaded out. I'm in trouble. I'm totally shaded out by other things around me. So I'm going to have to grow as tall as I can so I can at least get above the canopy at some point and start getting access to sunlight. And so that's why they get so tall. They're trying to get above the canopy and stop being shaded. And you can see that they didn't even really devote any resources into chlorophyll production. They're all yellowish. There's no chlorophyll going on. Their chloroplasts probably aren't even mature and ready to go. So they look pretty sad. Now, the good news is, if you do this to your plant accidentally or to your seeds, if you put them under a light or in your windowsill, they will green up. I mean, I suppose there is a point where they'll just die, but if you catch it early enough, you can turn etiolated plants into a more normal growth pattern. So the ones on the left were exposed to light appropriately, and you can see vastly different behaviors by the plant in terms of how they grow, what they're diverting their resources for. In the right, the etiolation, they're putting all their effort into, I got to grow tall, I got to get to the sun, I'm getting shaded. Whereas the ones on the left have all this luxury of, I got plenty of sun, I'm going to really develop my chloroplast, take advantage of that, do photosynthesis. Okay, so the fancy word for that is photomorphogenesis, where they will respond to light, morphogenesis, creation of form, changing. And so this photomorphogenesis just means that the presence or absence of light can do a lot of changes to the body form of the plant. Here is the idea of phototropism, which, if you've had Plant Kingdom, you've heard that term. Phototropism is this idea of plants, if they're getting access to what we call unidirectional light coming just from one direction. In this case, it's coming from the right and shining on this seedling. And it causes the plant to bend toward that unidirectional light. You see that if you have plants in your window, that they will bend toward the window, toward that incoming light. And so that's phototropism. And they've wondered, that actually got kicked off with Charles Darwin. He was very interested in why plants would do that. So he was the first person to really look at this idea. And subsequent researchers after him figured out that it's not sunlight in totality that is striking the plant, that the plant is responding to. It's actually the blue quality of light, of sunlight. And so, we know that sunlight can be broken down into the different wavelengths of light. And so when they just did a really simple experiment where they took these seedlings and just exposed them to different qualities of light unidirectionally. And it was only the blue light that allowed for the phototropism effect. And so we'll talk a little bit more about that when we get into hormone impact, because it's also, this sets off a hormone signal cascade as well. But they were able to match up this idea of the phototropism impact and blue light. So it matches up really well. And so this was good evidence to demonstrate that it's the blue quality of light that the plants are really responding to, not all the other qualities of light. So they've been doing simple experiments like that since, right around Darwin's time, Charles Darwin, in the mid to late 1800s. And the work has continued. They now know that plants can respond to light outside of what we call the visible spectrum. When we say visible, it's very human-centric. We're talking about what our eyes and retina can perceive. So the visible spectrum is 400 to 700 roughly. And we know that plants can see beyond 700 in the sense of they can pay attention to that quality of light. And then also down into the ultraviolet, that also will set off a signal cascade for them as well. So we know that there's photoreceptors that can respond to these different qualities of light. This is another famous experiment that they did early on, and these are lettuce seeds. And lettuce needs light. If you've ever bought a seed packet of lettuce, it says sow them lightly on the soil surface, don't cover them, because they do need sunlight in order to germinate. So here, the first top left, you see that they're in the dark and virtually no germination. Then you expose them not to the full spectrum of sunlight. They chose, let's just hit them with red light. And they germinated. So then they said, what if we put some seeds down, and instead of hitting them with red light, well, first hit them with red light, then go ahead and hit them with far red, and then let's see what happens. And when they did that, no germination. So then they're like, oh, okay, so the seeds are behaving very differently depending on if it's red or far red. So then they kept playing with it. They said, well, what happens if we go red, far red, red? And that's what's going on down here. So you can see that when they did that experiment and the last quality of light was red that the seeds saw, so to speak, they went ahead and germinated. And then the last one being shown here, they ended up hitting it with far red light. That was the last thing. And they didn't germinate. So the seeds were paying attention to what they last were exposed to. And they were responding to that information. And that got them hunting for what kind of photoreceptor might be able to pick up both red and far red light. So two close on the spectrum, but they're different wavelengths. And what they found after a lot of head scratching and a lot of research was this group of pigments that they call phytochromes, which we talk about in Plant Kingdom a little bit too. So these are interesting because they're photoreversible depending on if you hit them with red or far red light. You can get them to go into different states and give very different signals to the cells. So there's alternate forms of the pigment that they have designated. One form is called the P sub R. R stands for red, P for phytochrome. So the P sub R form of the pigment, if they call it the P sub R form because in that form, it's primed and ready to listen to red light. If you want to think of it that way, that's what it's going to absorb. So when it's in the P sub R form, it's ready to pay attention to red light. And when it does that, it actually shifts its form, its shape, to become the other alternate form, which is called the P sub FR form. Okay. So the P sub R, when it absorbs red light, it actually changes its shape. And then they start calling it the piece of FR form. If you have the piece of FR, FR is far red. If you put far red light on that version of the phytochrome, let it absorb that, it switches back to the piece of R form. It's like this toggle switch, back and forth, back and forth, back and forth. So you can play with that over and over, like what they did with those seeds. And so these phytochrome pigments are able, the piece of R form, it is able to absorb red light. Whereas the piece of FR form is able to absorb far red light. And you just get this interconversion back and forth. Going from the piece of R to the piece of FR, back to the piece of R. And what's happening is a plant that is absorbing red and far red light, what it's doing is it's actually kind of paying attention to the ratio of these pigments. How much of the phytochrome is in the piece of R form in my body? How much of the phytochrome is in the piece of FR in my body? That ratio is very important information for the plant because it provides information that allows it to do some really critical things in response to this information. Hopefully we'll get it to adapt to its environment, keep it alive, make sure it's doing its life cycle correctly and the timing of the life cycle correctly. And so the fancy way to say that is the ratio of the two pigments signals the plant and leads to appropriate shifts in the plant's phenotype, its behavior, its outward expression of its genetic information. Okay. So one important concept for this to make better sense is that when a plant is getting exposed to sunlight energy in the day, what you see is that the red to far red ratio, you get a little bit better red light coming at the plant. So it's 1.2 to 1. So slightly more red light is striking the plant body than far red. So keep that in mind. That's important information. Okay, so let's just look at this visually. This is another way to represent what we just talked about. Here you have the piece of R form, and they're trying to show that it got exposed to red light, and then it shifts physically into the piece of FR form. Now that form of the pigment, the phytochrome pigment, is capable of absorbing far red light, and if it does that, then it converts back to the piece of R form. You can also get the piece of FR to get converted back to piece of R just by long dark periods. So during the night, it will slowly convert back to the piece of R. The other thing to note here, which we'll talk about later, is that they're showing the piece of FR is the so-called active form, in the sense that very often what we see happening is if there's enough piece of FR, it will go into the nucleus and start interacting with proteins that regulate DNA gene expression. So the biological response, they're usually, what they're thinking of here is the piece of FR can go into the nucleus and start to impact gene expression. In a nutshell, that's what it's doing. Okay. So that's what they're showing on this next slide. And here they're showing the physical shifting going on. So the piece of R kind of looks like a M, if you want to think of it that way, like the letter M, and then the piece of FR looks like the Y. Kind of reminds me of YMCA dance. Okay, so you get this physical shift, and just that change in the shape causes this piece of FR form. It'll migrate into the nucleus and start, here you see protein-protein interactions. That means it's interacting with proteins that control DNA, gene expression, okay? And then if the piece of FR gets hit with far red light, well, then it converts back to this and you don't have that biological response. But really the plant is definitely, it's not just paying attention to piece of FR. They now know that it really is paying attention to the ratio of those two. And that ratio gives the plant really important information. All right, it's kind of a weird process, so spend some time thinking about that because it's a little odd, but we'll continue on with this on Friday, I guess, and talk a little bit more about some other photoreceptors too.